All right, so I just started passing back content check part 2. Let's see, the next content check is due Thursday. After today's lecture, you'll be able to finish most of it, and then after Thursday's lecture, you'll be able to finish it off. We do have a shortened time today, so please help me keep an eye on the clock so I make sure we're ending at the right time. Ending at 10:50. All of you showed up on time. That's fantastic. I was kind of worried about that because this election day schedule kind of throws off your habits a little bit here. I wanted to start off today with just some quick demonstrations about concepts that we talked about last week. So, make sure it pulls up here. Oh, let me restart this. I want to keep the camera on so that you can all see what I'm doing here. All right. When I did the content check, there are some questions about not understanding DNA wrapping, specifically like radial loops. So I have a string of beads here that I'm going to represent as a 30 nanometer fiber. All right, so DNA has been wrapped around nucleosomes, and they are in that zigzag formation. Right? I'm now going to start to twist up these beads, and it takes a little bit to get this going. And pretty soon, they'll get twisted up to the point where they start making these loops on their own. It's a little finicky here. Right, you can see it's already starting to bunch up quite a bit. And if I keep twisting, we're making even more loops. All right, and now I've been super condensed down to just this short segment out of what was once that really long bead of strings. So that's kind of how those radial loop domains work, right? By twisting it up, by making lots of loops, by making lots of twists, right? It really super coils, it shrinks down the DNA. And I could keep twisting and it would get even shorter. All right, so hopefully that visual helps. I also forgot to bring my string last time to show you a visual of supercoiling. So if we're talking about DNA replication, talking about topoisomerase, which relieves supercoiling, right? I mentioned that if you were to take a string and pull it apart, it gets knotted, right? And I wanted to show it to you, right? So if I pull this apart, it gets to a point where it's just like this knotted mess. I can keep pulling a little bit, but it's not going anywhere. All right, it gets all knotted up. It's so supercoiled that it doesn't unwind anymore. So this is what topoisomerase helps relieve, is this supercoiling as the DNA unwinds. All right, so hopefully that visual also helps kind of connect the dots a little bit. Right, I also saw in those content checks a little bit about, there's a lot to understand with DNA replication. There's a lot of different proteins. So I want to make one singular slide that might help you with studying. Right, so I have my image here. It contains almost all of the proteins that we talked about. We can break it down into 2 steps. Right, the DNA is going to unwind, which takes up the first 3 proteins: helicase, single-stranded binding proteins, and topoisomerase, all listed in my orange box here. And the other half of DNA replication would be the replication part, right? So first DNA unwinds, then DNA replicates. That's where we get all of our other proteins, our other enzymes coming into play here. We have the primase, which makes the RNA primer. We have DNA polymerase III, which replicates the DNA. We have DNA polymerase I, which is actually the one that is not shown on this image here. But that's the one that goes in, takes away the RNA nucleotides, replaces it with DNA. And then we have ligase, which glues it all together, right? And if we're thinking about replication, there's kind of 2 different ways this happens. We get our leading and our lagging strand, and it all has to deal with directionality. All right, so I know I just introduced this concept to you. I don't expect you to know it at the end of the lecture where I introduce it to you. I expect you to go home, study, Watch videos, draw this out, whatever you need to do to memorize this all, and then you'll all become pros. So it makes sense that it might be a little complicated now because you just haven't had the time yet to study this on your own. All right. Any other questions about what we've learned about so far? All right, before we dive into new material, I actually want to talk about that scientific literacy assignment. First of all, I've heard from several students that the login for the library isn't working well. I don't know what's causing that, right? That is something on technology services end that I don't understand. You should be able to use your regular login information. But if you're having a problem finishing that assignment, I will accept it late. Just send me an email that you did have an issue so I know to go back in and see that submitted assignment. Moodle doesn't tell me when things are submitted late. All right, second off, I just wanted to have a discussion about the assignment that you did, or at least completed most of. Question 2, in part 2, asked if you had any other questions about the peer review process. So we're going to have a shortened discussion about the scientific literacy assignment, and it's simply going to be me answering your questions about what you have with the peer review process. I've gone through this process a couple of times, right? So I understand it to a certain extent. So if you have any questions, now is the time to ask them. And I know you should have questions because it was part of the assignment.
How did somebody get selected to be like somebody who reviews it?
Yeah, good question. Sometimes the journal will reach out to like somebody in charge of a lab or a scientist doing work in a related field and ask them to be a reviewer. Sometimes they put out a call and people can volunteer themselves to be a reviewer. Reviewers typically aren't paid. This is a volunteer thing. They put their own time and effort to just make sure that the journals, the scientific information getting published is legit, it's scientifically rigorous, because we want our field to be rigorous. Yeah.
How did they prevent like personal bias in peer review? Because I was told a story by a professor where when they were doing a PhD, they'd have to go in front of like other people who have their doctorates, and they would be very harsh against him because they were competing against his professor, because they were all like trying to get grants and whatever doctor has the most successful PhD students, it looks better for them.
Mm-hmm.
So same thing like that peer being in the same field as you is competitive, right? So doesn't that cause problems in peer review?
It can. It's rare that it does in the peer review process. So I think the process your professor was describing was like his PhD dissertation and whatnot. Just different than peer review, but there's still like competitive edge out there. Some universities are very competitive in their research. They want the top minds in their labs, or they look down upon other labs that are not doing the same thing as them. So peer review tries to eliminate that bias by keeping the reviewers anonymous. That way they don't know who is being reviewed, and sometimes they will also make the authors anonymous as well, so they don't know who they are reviewing. Sometimes if it's a small field, you know exactly who it is anyways, because that lab might have a signature technique that they use. And if you're reading that paper that has that signature technique, you might be able to connect the dots, and that's where some biases come into play. Right? The goal is to eliminate biases, but there's always going to be some biases there, whether intentional or not. Right? We do our best to be unbiased as scientists, but being truly unbiased is never going to be a thing. Yeah. What other questions do we have? Like I said, I know you all have questions. Better open up your assignment and see what you wrote down. everyone in this room, were you familiar with the peer review process before this assignment? Yes and no? Okay. Well, I'll tell you that the peer review process is one of the critical parts of science, right? There are places where you can publish your paper and it doesn't need to be peer reviewed. There are journals that will accept just anybody. Typically you have to pay large amounts to get published in those journals. Those journals are not rigorous. I mean, you have to be careful with what is published in those journals. Because again, there's not been another set of eyes looking at it to make sure you're upholding academic honesty, to make sure you're not fudging anything, to make sure that your experiment is sound and that you didn't forget about some aspect of your project that you should have included, or that you're including the whole picture in your paper, right? So when you're doing research, it's really important to make sure that your articles have been peer reviewed. Open source articles can be okay, but they haven't been through the peer review process, so we just cannot guarantee it. Peer review process can take more time because of this, right? Sometimes it takes a couple of years for something to get published.
How would you go about like contacting someone if you had your own research paper? Would be like your, I guess, process and then contacting someone
Yeah, like, have you been published? Well, typically you'll contact a couple of journals and say, I have this paper, this research that I did, I would like to submit it to be published in your journal. And then the journal might contact you and say, great, let's get that peer review process started, and they will find the peer reviewers for you. Or the journal might say, I don't think you're a great fit for our journal, try somewhere else, and then you might contact another journal. Sometimes you have to contact multiple journals before it's accepted. Yeah. So it's not you having to seek out the reviewers yourself, which is great because we want to keep it anonymous, trying to avoid those biases. Any other questions we might have about the peer review process? All right, we're all experts on the peer review process then. If I had a question on the exam, which I won't put on, that asked you the peer review process, you would nail it. All right, since we're not a talkative bunch this morning, let's just move on into content. So today and on Thursday, we will be talking about cell division. All right, so we briefly introduced this last week. Today we're going to be talking about mitosis, and then on Thursday we'll move on to meiosis. So this might be a review for you, right? Maybe this is a good brush up on the basics. Maybe this is the first time you've really ever deep dived into it, right? But nonetheless, we are going to go through mitosis and meiosis. So a couple of slides to kind of get us into the mindset of why does a cell divide? What is cell division, right? Well, first of all, cells have to divide through pre-existing cells, right? We can't just spontaneously get a new cell out of nowhere. We can't throw in a bunch of atoms and molecules and enzymes into like a soup and then spontaneously have a cell, right? Cells are going to derive from pre-existing cells, right? And there's really two types of cell division. Like I said, we're going to talk about both of them. One is mitosis and one is meiosis. Now, when we talk about mitosis and meiosis, I'm typically going to talk about like humans or animals going through this process. And when humans and animals go through this process, more often than not, we're going to talk about diploid cells and haploid cells. Right? There are other organisms that might be triploid or tetraploid. They still go through these processes. Right? But I'm going to focus on diploid and haploid cells. So if we're thinking about mitosis here, right, in humans, we'd be going from a diploid cell to a diploid cell. So diploid, we have two chromosomes that are homologous pair, right? And we're going to end up with the resulting cells also being diploid. Meiosis, on the other hand, we're going to start with a diploid cell, and we're going to end with a haploid cell. So half the amount of the genetic material. Right? And we'll talk about these processes. No matter which form of cell division that a cell will go through, it's always going to end with this same process. This process called cytokinesis, which is what the image on the screen is showing us here. All right, so cytokinesis is when two cells physically separate and split apart. It's a splitting of the cytoplasm. That is the end result. We have cytokinesis, then we have two cells. All right, so we have mitosis and meiosis, and we know cells are going to divide from a pre-existing cell. Let's look just a little bit more into detail with the difference between mitosis and meiosis. All right, meiosis, we're going from a diploid cell to a haploid cell. So we're going to lead to the production of gametes. Right? So in most animals and many organisms, gametes will be called egg and sperm. Right? They are the haploid cells. Meiosis, right? We're producing these gametes. These gametes are going to be daughter cells that have half the amount of the genetic material. Because again, we're going from diploid to haploid. Right, mitosis is the opposite in that way. Mitosis is going to lead to daughter cells that are exactly identical. Right, so we're going from a diploid to creating more diploid cells. Now mitosis is not going to create gametes, but rather it's going to create all the other cells in our body. Right? All these other cells would be somatic cells. Right, so mitosis is creating these somatic cells. We're not creating gametes. We are not creating egg and sperm. The daughter cells are going to be genetically identical to the parents. Trying not to go too fast when we talk about mitosis and meiosis, because lots of terms can start getting jumbled together here. Right? Meiosis, gametes, daughters have half the genetic material. Mitosis, somatic cell, daughter cells are genetically identical. So we have these two different forms of cell division. We're going to start by talking and focusing on mitosis first. So why might a cell go through mitosis? Well, there's a variety of reasons. If it's a single cell organism, this is how it's going to reproduce. It's going to go through asexual reproduction. It's essentially going to divide its body in half and create an identical copy. If you're a multicellular organism, right, so you have many cells, you can still go through asexual reproduction using mitosis, right? So if you've ever had a plant and a part of the plant fell off and you were able to put that plant in another pot of soil and it grew into another new plant, right? So you propagated the plant. That's a form of asexual reproduction, right? It did not form gametes, it just simply kept increasing the number of cells in a part that had fallen off of itself. Right, now humans, we can't go through this. There are some organisms that can go through asexual reproduction. Right? You might have heard of instances in zoos where, I think monitor lizards can do this. They might lay an egg, but they were never mated with anybody. Right? That's another form of asexual reproduction. more often than not, though, if you're a multicellular organism, your cells are going to go through mitosis in order to grow the organism, right? So think of how many cells are in your body now compared to when you were a toddler, right? You needed to increase the number of cells in your body. You needed to grow. Or the repair of the organism itself, right? So maybe you broke a bone. Maybe you fell and scraped a knee and you have to replace those cells that you lost, right? So more often than not, mitosis is going to be for growth and repair. All right, so I've given you just enough background on mitosis versus meiosis to be able to answer this question. I want you to tell me which diagram is mitosis and which is meiosis.
The one that produces food is
Oh, yeah. I thought you meant looking at the third stage, and I'm like, no. I feel like she's. I wasn't sitting in the same room, sipping my coffee. Dog. You were here, you just didn't come. No, I wasn't. I call out about it 9:27. Oh. Yeah, I would have remembered that. If I would have remembered. I just can't believe that was the latest.
I have yours done already, so I'm going to give it back to you.
Thank you.
Is that your backpack?
Very long backpack. Great backpack. Because you can beat it all the time. Yeah, you don't have a backpack.
All right. I have given you two diagrams. Which one, the one on my right or the one on my left, is showing mitosis? Left. Yes, it is this one here. What made you come to that conclusion? What were the clues? Two daughter cells. What about those daughter cells? Yeah, they're also diploid, right? And the daughter cells look identical to the parent cells. Right? We've gone from a diploid cell to a diploid cell, and the daughter cells look identical to the parents. Whereas in meiosis, right, we have our parent cell, we have 4 daughter cells, right? But the daughter cells do not look identical to the parent. We'll go over meiosis on Thursday. We're going to focus on mitosis. All right, in order to start thinking about mitosis, we have to think about the cell cycle itself. All right, so the cell cycle is a series of events within the cell's lifespan that will lead to cell division. Right, so you kind of think of it as this cell's life history, right? It already knows its life history. It knows it's going to be one of these stages or go through all of these stages, right? So it's a series of events leading up to cell division. And these events, they're highly regulated. So these events are monitored and regulated. There are checkpoints along the way. We'll talk about some of those checkpoints. Right, but this is a highly regulated process. All right, so if we look at the cell cycle, right, this is a nice image of the cell cycle here. We can see there's different phases. G1 phase, G0, S phase, G2 phase, N phase. Let's talk about those phases. We're going to start with 3 phases that together are collectively known as interphase. Right? So interphase contains this G1 phase, this S phase, and this G2 phase. So if we're a cell, we'll start at the G1 phase. Right? We're a cell that's getting ready to divide. During the G1 phase, we are going to grow. Right? We're going to start growing in size. Right? We're getting ready to divide. We need to make sure we're big enough to divide. We will then enter into the S phase. Right? The S phase is where we will duplicate our DNA. This is where DNA replication occurs. Right? So that process that we talked about on last Thursday, right, happens in the S phase of interphase of the cell cycle. Right? So our entire genome is duplicated. Every single chromosome will be duplicated. The cell will then move on to the G2 phase, called the second gap phase. Right? Here we're going to have even more growth. Right? The cell is going to grow even more. It's also going to make sure that it's going to synthesize any proteins that it might need for the process of cell division. All right, so collectively these 3 phases are called interphase. We have the G1 phase, the cell grows. We have the S phase, DNA replicates, and the G2 phase, the cell grows some more and synthesizes any protein it might need for division. All right, now I like this image showing interphase here, because if you were to look at interphase and try to pick out a singular chromosome in that nucleus, it would be really hard to do, right? Because during interphase, the DNA is duplicated, right? But, well, just lost my captions there. It's not yet condensed. So if we were to look at a cell in interphase, it would kind of look very messy. I refer to it as it looks like a plate of spaghetti, right? It's hard to distinguish the noodles apart from one another. All right. Oh, and I forgot I had another image up here. All right, so remember during S phase, we're replicating our DNA. This is where we would go from a single chromatid to having sister chromatids, right? So keep that in mind. We'll be talking about sister chromatids and how they split during mitosis. Oh, well, captions don't want to show up for me anymore. We'll have to go the rest of the lecture without captions. Don't know why that is. All right, so we've gone through interphase, right, which is the first 3 steps of the cell cycle. But there are 2 other phases during the cell cycle that this cell can be a part of, right? After interphase, right, it's getting ready to divide. It is ready to divide. It's going to move on to the M phase. And the M phase is where cell division actually occurs. This is where mitosis and cytokinesis, so the splitting apart of the cytoplasm, actually occurs, is during this M phase. All right? Now we'll go in more depth on the M phase here, but for now we're just going to say this is where mitosis occurs. It could be that we have a cell that isn't going to divide. Maybe it's never going to divide. Maybe it's not going to divide right now. Right? So it does not need to go through interphase and the M phase. Instead, it's going to hang out in what's called the G0 phase. Right? This G0 phase is kind of this offshoot here, right off of the G1 phase. It's its own little cycle. Right? So if a cell is never going to divide, if a cell doesn't need to currently divide, it's going to hang out in this G0 phase and do exactly that. It's just going to simply exist. It's going to perform its functions. It's going to exist, but it's not going to be actively preparing to divide. All right, so we have interphase. We're actively preparing to divide. That includes G1, S, and G2. After interphase comes the M phase, where it is actively dividing. And if it's never going to divide, or if it's going to divide later on, not right now, we'll have the G0 phase. All right, so what determines whether a cell is going to divide or not? Right? How does the cell determine that, yes, it's going to leave the G0 phase, enter in the G1 phase, and start interphase? Right? Well, it depends, right? There's a variety of factors. They could be external factors, so factors beyond the cell itself, right? Maybe it is that you fell and scraped your knee, right? Now those cells need to replace those lost skin cells, right? So maybe that's the factor that starts cell division. Maybe it's certain signaling molecules signaling to the cell that we need more cells, right? Maybe this is like a hormone. Maybe it's like a growth hormone. We need more cells, right? Any type of signaling molecule indicating that more cells are needed here. So it could be those external factors. It could also be internal factors, so within the cell itself, right? So the cell that is in this cell cycle, whether it's in G0, so just kind of hanging out, or actively in interphase, right? Or it's just starting interphase here. There are some internal factors telling this cell whether or not it should divide, right? So there are control molecules within the cell cycle, right, that tell the cell, yes, go ahead, or hang on, we don't want to divide just yet. There are also checkpoints. Right, so within the cell cycle, there are checkpoints, kind of like a security station. Make sure that the cell has the proper credentials before it can move on to the next phase. Right, so a whole variety of factors determining whether or not it's going to divide. Once it has determined it's going to divide, right, it's going to proceed through the cell cycle. And like I said, the cell cycle is highly regulated. So within the cell cycle itself, as it goes through the cell cycle, there are checkpoints to make sure everything is okay for this cell to proceed with cell division. Right? There's actually 3 checkpoints in eukaryotes. We would say it's the G1 checkpoint, the G2 checkpoint, and the metaphase checkpoint. So metaphase is a phase within the M phase. And if we look at this image, it kind of gives us a very brief description of what these different checkpoints are looking for. Right? So the G1 checkpoint is going to make sure that this cell has proper nutrients, it has proper growth factors, it's ready to grow, and to make sure that this cell does not have any DNA damage. Right? We don't want a cell that has damage to its DNA to replicate that damaged DNA and pass that damaged DNA onto its daughter cells. Right? So we're checking for correct DNA at this point. The G2 checkpoint, right, so after DNA has replicated, is going to make sure that everything went well in DNA replication. That the DNA has replicated, and it has replicated properly. There's no major mistakes. And it's going to make sure that that cell is ready to divide. It has the proper cell size. It's grown enough. All right, so if it's past this checkpoint, it might get to this metaphase checkpoint. And this metaphase checkpoint is going to make sure that the chromosomes are properly attached to the mitotic spindle. We'll talk about that in a second. But they're properly attached within the cell. That way, when cell division occurs, they will properly split. Right, so these checkpoints, they are these sensors. They are making sure that this cell is healthy, ready to divide. If it's not healthy, this cycle can be delayed or postponed indefinitely. Right, maybe we'll never let this cell go through cell division. It's when these checkpoints start to fail that we can get things like mutations or cancers starting to occur. Right? So these checkpoints, they do play a very critical role within our cells, within our entire body. Yeah?
What happens if say the checkpoint that checks if the cell has grown enough? What if that fails? Like, what happens if the cell divides when it's not big enough?
When it's not big enough? Likely that means it hasn't produced the right proteins so that if it does go through cell division, it's going to be halted somewhere along the lines. Like there's just not the right proteins for it to proceed. Cell division can get really messed up. That's likely what's going to happen. All right, let's do a quick check-in. So I want you to tell me what are the 5 stages of the cell cycle and what happens in each stage. I'm actually going to tell the class that one, yes. Wow.
I feel like Miranda might have known that you were like
You're the only one.
I know.
Or well,
5 stages. But they made it 5.
Oh yeah, you're right.
I know what
I did not.
So it's G1 grows.
How was your weekend?
G2.
Fun. I was
And
for my
workshop. That's what my mitosis is. It's like the first one you grow, the second one you
also
size,
I
the
have
third one
a
you
second
size.
workshop now. They have my hours. You know, I work 7.5 hours a week.
you need to be a worse employee.
Yeah. But
They're making me work from 10 a.m. to 2 a.m. on Friday.
Oh, okay.
Campus G1, SB, G3. It was a guarantee. What are the chances someone calls me and is like, my friend's having a heart attack, and I just fuck them up. You're in 1A.
All right, my timer stopped when I put the captions back on. But I think it's about 2 minutes. So let's move on. What are the 5 stages of the cell cycle? What is the first stage? G1. What happens in G1? Growth. All right. What's the second stage? S phase. What happens in the S phase? DNA replication. The next stage? G2. What happens in G2?
More growth.
More growth. We're synthesizing any proteins needed for cell division. Right? And then after these 3 phases, which are collectively called interphase, what comes next? Yeah, mitosis, the M phase. So we're actually going to go through cell division. Now, if a cell were to never divide, what phase would it enter into? G0. Yes. All right. So at this point, we've talked about our cell cycle. We know how the cell gets ready to divide. And we know that division happens within our M phase. Right? Let's talk about this M phase a little bit more in detail. All right, so our M phase, this is where division actually happens. And in mitosis, the division will result in 2 cells, 2 new cells, that are identical to the original cell. Now, I've already started using this terminology But often I'll say that the original cell is the parent cell or the mother cell, right? And the resulting cells are the daughter cells. Right? That's pretty common terminology when we're talking about DNA replication, cell division. All right, so mitosis has all of these steps involved, all these different phases. We have prophase, we have prometaphase, we have metaphase, anaphase, and finally telophase. Right? So on an exam, it is very likely that I might say, I have this cell, right? These are how many homologous pairs I have within the cell. I want you to draw this cell going through anaphase. Right? So you'll have to know what's going on in each of these phases, and you'll have to know which order these phases come in. Right? Now I know there's a lot of different ways to remember this, and if you Google it, you'll probably find a bunch of like short sentences, like where the first letter of each word is the first letter of a phase, right? I like to remember this as you just adopted a new puppy. The puppy is not potty trained yet. You're going to be using a lot of PP mats, right? That's how I remember it. That's what sticks in my mind. Maybe one of those phrases you Google will stick in your mind better, right? But that's how I remember it. All right, let's go through these stages, and we're going to go through these stages one by one. And if you've learned this already, it might feel a little tedious, right? But keep in mind that this is going to be on your next exam, so this might be a really good refresher. We're going to start with our first phase, which is prophase. And I'm actually going to draw these phases out as we talk about them. All right, so prophase, we have our cell. Chromosomes in prophase are starting to condense, right? It no longer looks like a plate of spaghetti, a plate of noodles. Now it looks like chromosomes, like I can pick out each individual chromosomes, right? So I'm going to draw my chromosomes that have been condensed. Now I'm going to draw them as X's because I know they've gone through DNA replication at this point. That happened during interphase. And I'm going to draw two homologous pairs in my drawing, right? So I'm using color to indicate which one came from mom, which one came from dad. And size is indicating homologous chromosomes. So these small ones are homologous to each other, and the big ones are homologous to each other. All right, so my chromosomes, they're condensed. The nuclear envelope, remember that's where DNA is housed, in the nucleus. The nuclear envelope, during prophase, during mitosis, is going to start to disappear. So I'm going to draw it as a dashed line. That nuclear envelope is starting to disintegrate. It's starting to disappear. All right, so our chromosomes are condensed. Our nuclear envelope is starting to disappear. We have one more thing that is happening here, and that's this thing called the mitotic spindle is starting to form. So I'm going to draw it on my drawing, and then I will explain what the mitotic spindle is. So I'm going to draw it as two dots with some lines starting to come out of it. All right, so mitotic spindle has started to form. What is the mitotic spindle? Well, the mitotic spindle is what coordinates the chromosomes moving during cell division and coordinates them splitting during cell division. So the mitotic spindle organizes and sorts our chromosomes, and it's composed of two parts. It's composed of the centrosomes. Right, so in my drawing, I drew the centrosomes as a dots. On this image, the centrosomes are these yellow dots here. The centrosomes can often be called the microtubule organizing centers, like it is in this image, the MTOCs. Right, I think it's easier to remember centrosomes. If you told me MTOC, that would also be correct. Now the centrosomes are protein complexes, and they have duplicated during interphase. So remember during G2, that cell is growing, but it's also producing any proteins it might need in order to replicate. So during that G2 phase, these centrosomes have been replicated so that there are 2. All right, so the mitotic spindle contains the centrosomes. It also contains microtubules. Now that should be a term that you recognize. There are microtubules within the cytoskeleton of a cell, but microtubules are long protein filaments. Right, so if we're looking at our image up here, our microtubules are these long green lines on my image. Right, so when I drew it in prophase, I started drawing those lines radiating out from the centrosomes, right, because they are starting to be formed. All right, so we're still talking about prophase at this point, right? Prophase, nuclear envelope has started to disappear, chromosomes have condensed, and the mitotic spindle has started to form. So we're going to move on to our next phase, which is prometaphase. I'm going to label my first one here. Right now, especially if you learn this maybe in high school that used an older textbook. Some older textbooks do not consider prometaphase an official phase, right? But nowadays it's more common to include it as the official phase. So we are saying that this is an official phase, all right? So you might see, like if you Google images of mitosis online, you might see photos of this process that exclude prometaphase, right? But I want you to know prometaphase. So during prometaphase, the nuclear envelope is completely gone. Right, so I'm not going to draw anything in my drawing for the nuclear envelope because it's completely gone. My centrosomes, right, are on opposite sides of the cell. I would say it's at the 2 poles. If we think about planet Earth, right, we have 2 poles. We have the North Pole, we have the South Pole. They are on opposite sides of the planet. Right, so if we think about a cell, the poles would be opposite sides of the cell. My centrosomes are on opposite sides of the cell. So I'm going to draw that in. They have now migrated to be opposite one another. Right? Now I'm going to draw my chromosomes. They're still condensed. But what's different about the chromosomes is that the mitotic spindle is completely formed, which means the microtubules are completely formed, and the microtubules are attached to the chromosomes. In fact, they're attached to the chromosomes at this place called the kinetochore. Right, so the kinetochore is the physical location where these microtubules attach to the chromosomes. So I'm going to draw it on my drawing here. I'm going to draw from each centrosome. It's going to have a microtubule attaching to either side of every chromosome. All right. So my mitotic spindle is fully formed, and the microtubules are attached to my chromosomes. All right, so prophase, nuclear envelope starts to disappear, chromosomes condense, mitotic spindle starts to form. Prometaphase, that nuclear envelope is completely gone, the mitotic spindles are completely formed, and they're connected to the chromosomes. All right, after prometaphase, we get metaphase. I like metaphase because I think it's the one that you can look at and very easily distinguish from the others. Because what has happened in metaphase our chromosomes are lined up in the middle. All right, so I'm going to draw my cell. My mitotic spindles are completely formed. The centrosomes are still at the poles. My chromosomes are lined up in the middle in a singular row. Okay, and the mitotic spindle is still attached to these chromosomes. Right, so it's the mitotic spindle that's actually aligned to our chromosomes because they were attached in prometaphase, and now the mitotic spindles are tugging and pulling, right? They're constantly adjusting until those chromosomes line up directly in the middle. Now, when those chromosomes line up in the middle, we would call that the metaphase plate, right? The middle being the metaphase plate. The metaphase plate is not like a physical item that you could touch, right? It's just a zone within the cell. We call it the metaphase plate. It is the area in which they line up. All right. We'll label this one here. All right, so let's review the first 3 stages, make sure you know what's happening, and then we'll move on. Might be helpful for you to draw this out or redraw it if you have been drawing it out. Mostly review.
Joyce here, hold on.
It's gonna be such a
Yeah, it tends to be mostly review, but
Yeah.
it'll be important when we talk about services and
Tiny cells. Well, cells are small. They should be small. What's your favorite tiny teeny?
here.
I know it.
Tiny teeny thing. My rabbits.
Yeah. Yes.
I just told myself that next week I'm gonna have to study so much.
Yeah.
Around processes.
So this comes up around me twice a week, so I throw a lot at you at once. I have bad eyes and a lot of black.
Can you not tell
Or
me
have
that?
candy toys.
No.
It's not that easy, guys. That way,
Not this Friday.
Trying to get the full effect.
It's a Tuesday this time.
Oh,
Yeah,
okay,
better
so it's
to
not
get
next
it.
Friday, but it's the Tuesday after next Friday. Like, wait, or is it 2 more?
Can you get the one more back on Tuesday?
There we go.
So the only difference between promethazine and Tuesday is the line that line.
Yeah.
Oh, I love this photo of her. Look at her. Oh, but all the way. That's where you get it.
I think it started this time.
Started making it one. Wait, so it's time.
It's crazy that you only have one.
When you go to a really small school,
Well, the end you
when you
really
really
can't get
And you go to the second spot.
that's fun.
Is it kind of like a small business?
We
Like
also
there's
lost our
like
chemistry
you in
teacher in 10th grade.
So out of
I forgot to set my timer, but looking around the room,
No science for you.
it seems that we finished up, so let's talk about it. Let's see, stages of mitosis. What is the first stage? Prophase. Oh, we did. Correction. There we go. Prophase. All right, and so what's happening in prophase? Right, chromosomes are condensing. Nuclear envelope is falling apart, and the mitotic spindle starts to form.
Hey, is that Oh, it's over.
What's the next phase?
Pro-meta.
I didn't get it.
Prometaphase. What's happening in prometaphase?
They're colliding.
Mitotic spindle has moved to the poles. The nuclear envelope is completely gone. What else?
The mitotic spindles form like these fibers that grab onto the chromosomes.
Yeah, so mitotic spindles, not only have they migrated to the poles, the microtubules are fully formed and they are connected to the chromosomes. All right. The last phase that we talked about so far. Metaphase. What is happening in metaphase? Yeah, the sister chromatids, all the chromosomes, are lined up in a single line. What do we call that single line? That area? Yeah, the metaphase plate. So chromosomes are lined up on the metaphase plate. All right. Feeling good so far? Ready to move on?
Oh, wait.
All right, let's go through the last few stages of mitosis.
There's more?
There is more. Now I noticed in the content checks that a lot of you want like one image that shows everything that's happening. It's really hard to find. But when I do find it, I'm going to try to add it into my slides, which is what this slide is depicting. Right? This is depicting those 3 stages we just went through so far. So I'm not going to talk about it too much, but it might be helpful when you study. All right. On to our next phase, which is anaphase. All right, my piece of chalk has disappeared here. So by the time we're at anaphase, right, the chromosomes are lined up on that metaphase plate. So when anaphase happens, those chromosomes are going to start to split apart, right? Because remember, our chromosomes consist of 2 sister chromatids, right? And the sister chromatids are identical to one another. Those sister chromatids are going to split. So let's draw this out. That was a little wonky there, but that's okay. All right, so I have split my chromosomes. All right, my sister chromatids, they have split apart. They are no longer connected at the centromere, at that joining part of the chromosome. Now my mitotic spindle is still attached to these sister chromosomes, or sister chromatids. And this mitotic spindle has started to shorten, right? It's shortening. And the shortening of this mitotic spindle is what causes our chromosomes to split in half. So in anaphase, what we see is 2 rows, essentially, of chromosomes being moved to either side of the cell. They're being moved towards the poles, right? So they're moving towards the poles. All right, still don't have a nuclear envelope. Mitotic spindles have started to shorten. And what's key here is these sister chromatids have split. All right, now notice we haven't split apart homologous pairs yet. Because if we notice what's going to either side, right, we still have 2 small and 2 big. We've just simply split apart the part of the chromosome that's been replicated. Right, so we still have homologous pairs. So then our last phase here would be telophase. It's not the last step of mitosis, but it's the last official phase. So if I were to draw out telophase, actually going to draw it as one big cell. By the time telophase is happening, our sister chromatids that have been split apart are now fully moved to the opposite sides of our cells. So we'll draw that in. They are fully moved. And then during telophase, the nuclear envelope starts to reappear. So that nuclear membrane starts to reform. So I'm going to draw that as 2 dashed circles. So essentially now what we have is one big cell with 2 nuclei. Each nuclei containing the same genetic information. Each nuclei being identical to one another. All right, the chromosomes, once they've reached their poles, and once that nuclear envelope starts to reappear, they're actually going to start to decondense. Right, so I've still drawn them condensed. If I were to draw this as later on in telophase, I would draw them more string-like, more spread out, right? Because they are starting to decondense here. All right, but this isn't the last step. Right? It's the last phase, but it's not the last step. Because we don't want one cell with 2 nuclei. We want 2 cells, each with their own nuclei. So we have to go through cytokinesis. All right, so I'm going to draw a cytokinesis on this sideways figure 8. Right? So what I'm drawing here is what's called the cleavage furrow. Right? It's starting to furrow. It's kind of like if you were to put a string around the center of this very large cell and start to pull, right? It'll start to cinch it. Right? So we have what's called this cleavage furrow. We're starting to split our cell into 2. And again, each cell still contains these chromosomes. Actually, it would be more appropriate if I were to draw these chromosomes totally decondensed, because at this point they are. So I might want to draw them more spread out. All right, so this would be the process of cytokinesis. So by the end of cytokinesis, I would end up with 2 individual cells. Right? They would each be on their own. All right, so now we've gone from our parent cell to 2 daughter cells, and the daughter cells are genetically identical. Right? So if we look at the cell that entered prophase, right, this is after DNA replication, but we have 2 homologous pairs. Right? We still have 2 homologous pairs. Now, if these cells were to go through replication, DNA replication, they would look exactly the same as what we started with in prophase. Right? But we've started with a diploid cell, we have ended with a diploid cell. 2 diploid cells. Now, cytokinesis happens differently in plants versus animals. Right? So in animals, we get this cleavage furrow. We can kind of take a drawstring analogy and like cinch it apart until it completely splits. We get that cleavage furrow. Now plants, remember, they have a cell wall, and that cell wall is very rigid, right? So we can't just cinch apart that cell wall, right? It doesn't work that way. Instead, the cell is going to create a cell plate. And it's very faint on this image, the way the light is in this room. But if you look closely, there's this line starting to form in the middle there. Right, so the cell plate will form, and eventually that cell plate will become the cell wall surrounding 2 different cells. All right, let's do a quick review I'm gonna give you about like 5 minutes here, which is kind of short. But I want you to look at the next 2 slides. Review what's happening in each slide, and then tell me which cycle, which phase each of these cells are going through. And then we'll review before 10:50 our class period is up. Yeah?
When it comes to like plants,
Yeah.
are the cells going bigger afterwards? What's preventing them from being?
Sorry, I was doing the.
Yeah. So I mean, they will start out as 2 smaller cells, but then they can grow. They can add in more cytoplasm to expand their plasma membrane. Yeah, that cell wall can grow with them. Yep.
So just cell wall it takes too much energy to like situate. That's why.
Yeah, it sounds like flexible as like an animal cell. It's more rigid, so it's harder to situate. We just have to form like a plate.
Oh, no.
We'll divide it. Yeah.
But it can stretch.
It can't stretch, but it can add more to its cell wall to make it longer.
I don't know what is going on.
So it has to. It's a lot more energetically excited.
Maybe 4 is.
Right. So I don't know, for some reason my brain is thinking about how some crustaceans like shed skin.
Oh,
Is one
yeah.
I'm sure that's not what plants do, but
Yeah, I'm trying to like figure out how I could like weave that in, but
I don't care.
it's kind of hard. Yeah.
I mean, like some like seashells do this. Like they form like they basically add their like to the size of their shell. Yeah, sometimes they pour ducks. You can tell the line where each attempt at making their.
I don't have a turtle. What biology?
Oh, I think bean, because my aunt has a turtle.
I don't know, I feel like 5 has like where it's that equally. Like this one is, there is no.
How the fuck is that?
Like,
It's named Ian. Maybe it's named that. No, I just said it might be Bean.
I'd say 5 is.
Could be, I don't know. Okay.
The nuclear membrane cells, then the spindle forms, and then the spindle attaches to the chromosome. And then the spindle, does the spindle line up the chromosome? Yes. Okay. And then
They move them to
the middle onto the plate, and then they divide the plate. They divide those.
then they rip them into.
And then they start to form the nuclear thingy again. And then they become little balls. And then once they're little balls, then they divide.
Where they do the same thing.
Easy peasy.
So until they, it's like, what?
I mean, yeah.
People, you know, when I'm talking to people, that usually it's like I was looking at them, you fucking I'm over here growing television. If you really think about it, Anapace is like a monkey holding 2 the
What is the whole cell got DNA? There's no nuclear membrane. It's not the end because that's splitting. Like the way of 2 like 7
If you look at that image, could you potentially tell the 4? It's got only 4 little tentacles.
No, it has way
It
more.
would be hard,
Yeah.
but it's much easier than say number 5.
Yeah.
So there, it's telling you it's the chromosomes are starting to condense.
Oh, so you're saying it's sort
How
of.
many chromosomes are there?
Oh, because each cell will have.
Are you saying like that? Because like the thing is like I wouldn't be able to tell like the nuclear membrane out of that. I feel like. Unless maybe that's because it's a circle. That's what it is. I
The
don't.
nuclear membrane is going to be really hard.
One tentacle.
So you'll have to rely on what chromosomes are.
How many
Oh.
How many chromosomes are dispersed?
Oh, that are broken. Got it. Okay. Thank you.
I know there's a number that we know about.
And it's not 4.
It's not 4.
So every cell will have all the chromosomes.
See, that doesn't make sense.
Why does that not make sense? I only know medical things.
Let's go with a bot.
You know, genetically it
Weird to have it. It's like we just have to.
5 is broken.
Okay. Maybe
Because it
not.
starts off as
Or
a
sorry, 1 is broken.
I didn't want to say that. That's too different. I had to. It's fine. One prophase. I do. I do.
Some people don't know.
Oh, bro, metaphase and stuff. I really want to talk about
It is.
So it's like all what does that do?
I was in front of it.
No, I don't know anything. It's so take my word for it. I don't know. You don't have to go to the giant. That's what happened.
Because I woke up at like 5:45. Quick, go back.
He was twice a minute.
He's in my favorite movie.
My dad used to be able to quote every line in that. Yes. I read. Have you read the book? I read it when I was like in 4th or 5th grade. It's very good.
Very good for the 4th or 5th grader, or very good for
Very good for now. It's a book for adults. Not for 4th or 5th. I shot it. I just was very good. Maybe it isn't. I cannot wait to have so much free time with it. No.
Yeah, I see. I requested a mail-in ballot, and then,
I don't really have enough information, so
like, I
I don't feel reasonable for doing actual information. Just because I'm not very aware.
you're supposed to spend 5 minutes taking pictures of it.
No, I'm not very aware, and I need to have like lots of background information.
All
I'm
right,
gonna put
let's
this
go over these answers so we have time before class ends here. Our first phase, prophase. What is happening? The mitotic spindle is forming. Nuclear envelope is disappearing. Chromosomes are condensing. Yes. All right, so we move on to our next phase, pro-metaphase. What is happening? Is there a nuclear envelope? No, nuclear envelope is completely gone. What happens to the mitotic spindle?
Opposite poles.
Yeah, they're at opposite ends, opposite poles, and what else? Yes, and the microtubules are attaching to the chromosomes, right, at that kinetochore, that specific part on the chromosome. All right, so our microtubules are attached, they're fully formed, they're at the poles. What is happening in metaphase? Yes, the sister chromatids are lined up on the metaphase plate. All right, so they're all in a line. All right, then we move on to anaphase. What's happening here?
Sister chromatids are getting pulled apart.
Sister chromatids are getting pulled apart. All right, so this is where it's really easy to say the chromosomes are being pulled apart or they're splitting. But I want you to specify that it's the sister chromatids that are being pulled apart. All right, they're being pulled to the poles. All right, and telophase and cytokinesis, what is happening here? The nuclear envelope begins to reappear. Chromosomes are starting to decondense. And cytokinesis does what? Splits them apart. Yes, splits that cytoplasm. We are ending up with 2 daughter cells. All right. Any questions about these phases? All right, let's put this into practice then. So, I have a variety of cells circled that are in different phases of the cell cycle and in mitosis. If I were to ask you, number 1, what phase do you think it is in? Would you have a guess?
Prophase.
I would say this one is in prophase. Right, so the clues that led me there is that compared to most of the other cells here, these chromosomes look like they're starting to condense. I can start to make out individual chromosomes. Now it'd be really hard to completely take them apart, so they're probably still decondensing, or it's still condensing, right? But I would say this is entering into prophase. All right, what about number 2? telophase. Right? It looks like I have one cell with 2 nuclei, and those 2 nuclei are starting to decondense. Right? Now, if you look really closely, you could argue that this is just a picture of 2 cells on top of each other. Right? Could be that too. Right? But if we assume that it's one cell with 2 nuclei, it would be telophase. What about number 3? Yeah, likely metaphase. All those chromosomes are in a line, in a row. Number 4, anaphase. They're starting to split apart, and we have essentially 2 rows moving to opposite ends. What about number 5? What was that? Yeah, this is likely interphase. All right, so this is a tricky one. So if I look at the chromosomes, they are not condensed. Right? It looks like a plate of spaghetti, a bunch of noodles all put together. This is likely a cell in interphase. Now, what phase of interphase? It's hard to say, right? But it is in interphase because the chromosomes are not condensed. All right, what about number 6? What was that? Yeah, there could be a variety of answers here. This is a little hard to tell. I put it as pro-metaphase, moving into early metaphase. Right? It looks like they're starting to move in a row. You might have said that this could have been the start of anaphase. Maybe it looks like they're starting to split apart. They're not in a nice, neat row, so it's probably one of the phases on either side of the nice, neat metaphase. All right. Again, I have this synthesis slide for you to look at and use to study. Right, there's that assignment due tomorrow, as well as the content check due on Thursday. The assignment tomorrow should be really short and easy. Easy 5 points. It's more like the people involved with the discussion.
Dude, I could discover the most important thing in the world, and I don't think anyone talks about it the second I die.
What if somebody else took credit for your discovery?
I'm dead. I don't care.
Oh, your family will get the
Yeah,
money.
your family wouldn't care.
Watson and Crick
I'm
steal
dead. I'm not
credit.
gonna know.
Yeah, I don't know. Well, the thing is,
Didn't Watson and Crick steal credit?